I'll be your host for today's event. I'm Jeff Bogaliscus, technical editor for Genetic Engineering and Biotechnology News, which for the last 40 years has been delivering the latest news on the most innovative topics within the life sciences industry. Now, I'm sure many of you in the audience know the rigors of DNA library preparation for next generation sequencing. It can be a long, multi-step process, yet it's integral for proper sample management and reproducible results. But there is hope for simplification, as automating this process can reduce costs, free up researchers' valuable time, and streamline laboratory workflows. And one area where this automation step has become extremely advantageous is vaccine development. Now, the emergence of novel infectious diseases and outbreaks of well-known communicable agents has underscored the increased demand of vaccine candidates for humans and livestock. As such, products or equipment that can make NGS preparation and analysis more straightforward and effective are a welcome addition to any laboratory. And in today's webinar, we're going to hear about the use of an automated library preparation system in the development of vaccine candidates. But before we jump into the webinar, let's first meet our speaker for today's event. It's my pleasure to welcome Dr. Graham Freimanis, who is a research scientist and head of high-throughput sequencing at the Pirbright Institute in the UK. Graham is a trained virologist and an expert in the use of genomic applications for the investigation of veterinary viruses in livestock. Now, Graham has been at the Pirbright Institute for over 10 years now, where he's focused on using genomic technologies to dissect the role of viral evolution and diversity in veterinary viral disease. Now, today, Graham is going to show us some data from his recent work where he compared data from sequencing-ready libraries generated using both manual workflows and the new Tecan MagicPrep NGS automated library preparation system, using samples from several species of economically important livestock inoculated with different vaccine candidates. Graham, over to you.
Hi, my name's Graham Freimanis. I'm head of high-throughput sequencing at the Pirbright Institute in the UK, and I'd just like to thank Tecan Genomics for giving me the opportunity today to talk about the beta tests we've been performing and to talk about how one of their new high-throughput sequencing library preparation machines we've been testing has played out in the lab. And the MagicPrep, as it's now released, we've looked at how it performs and integrates into our RNA and DNA sequencing workflows and looked at how it compares with some of the other kits that which we've been using in the lab. So the Pirbright Institute is based in the UK. It's about 40 miles south of London. The Institute is a world-renowned centre of excellence for veterinary viral disease of livestock, and it focuses in particular on viruses of transboundary and economic significance, which primarily include zoonotic infections such as African swine fever, foot-and-mouth disease virus, and Newcastle disease virus, among others. We operate in SAPU4 high containment environment. Our SAPU4 building is in the top right-hand picture, and the Plowright building. With most of our endemic viruses we work with, such as infectious bronchitis virus and influenza, avian influenza and so on, are in the containment level 2 building, which is shown in the bottom right picture. We also have facilities on site for rearing in insects in the insectaries, where bioimaging, high throughput sequencing and ACDP containment level 3 laboratories facilities. So the Institute's remit really is to provide the UK with a capability and a capacity to deal with animal infectious disease. And we try and do this through strengthening the food security for the future in the UK. And we contribute mainly to this through 4 main areas. These include epidemiology, diagnostics, basic research, and developing new vaccines for animal viruses. Our specialist expertise and research interests fall into areas of veterinary viral diseases and livestock, as I've said in the last couple of slides. And this is reflected in the role that we also play in surveillance, particularly in collaboration with the World Organization for Animal Health, formerly in the OIE, and their reference laboratory networks. We currently hold 10 reference laboratory World Organization for Animal Health laboratory status per right, and these form an important part of these collaborative networks, including 2 world reference labs for foot-and-mouth disease and rinderpest. So why is animal health research so important? In as early as 2013, 13 zoonoses alone was thought to be responsible for almost 2.2 billion human illnesses a year, or 2.3 million deaths. This was more recently expanded to almost 61% of human pathogens zoonotic in nature, and this is expanded across bacteria, viruses, protozoa, parasites, and other pathogens. And in 2019, the UN forecast that in 2050, the global population was going to reach 10 billion people. And this would place an unprecedented and extraordinary pressure on our current capacity to generate food and capability for agriculture. And this was expanded by a study by Kelly et al. In 2020, which analysed our current food production capacity, and suggested that that may have to increase by up to 70% to cope with this. Therefore, prioritising animal health and the health of livestock and so on is of a top concern for most policymakers now in the world globally. As a technology, genomics has undergone a radical emergence and transformation over the last decade, making it significantly more accessible to a large number of labs. These are mainly through the increased availability and cost effectiveness of benchtop sequencers, establishment of core facilities, and so on. And as an example, in 2012, almost 1,000 human genomes had been sequenced, whereas by 2020, this number had exceeded 1 million. These days, this is all in the backdrop on context of the recent COVID-19 outbreak and pandemic as well. And we look at the role that genomics has played within the viral surveillance of the outbreak. And these days, high-throughput sequencing is routine in pretty much all areas of society in terms of academia, medical and industry environments. Genomics now plays a central role in animal health too, both in surveillance, basic research, and vaccine and drug development. It drives a better understanding of host immunity and life cycles of those viral pathogens themselves and how the two interplay with one another. And with the increased availability of high-throughput sequencing, both the technology and the increasingly diverse applications that it's able to perform, high-throughput sequencing now is increasingly more suitable and employable to research in animal health. However, one of the main bottlenecks in high-throughput sequencing was identified by a study by Tyler et al. In 2017, is sample preparation. So this was identified in that study as a time-consuming, a tedious, repetitive, and error-prone component of the pipeline. Other studies in 2017 also looked at the rapid turnaround of diagnosis in urgent cancer pipelines for turnaround of results back to doctors for decisions in preclinical and postclinical. And these identified that those pipelines with molecular and genomics components, the sample preparation was the biggest bottleneck. And time and cost still remain the biggest barriers to a more widespread use of genomics in today's society. Automation is a great way to resolve and reduce some of these limitations. And some examples of current high-throughput sequencing protocols and their subsequent automation saving hands-on time include that of the NextZero-XT workflow, where up to 96 reactions on an adjuvant Bravo have been automated to reduce hands-on time from 8 hours down to 25 minutes. A PerkinElmer DNA library prep performed on a cyclone. A PerkinElmer cyclone has been reduced from 4 hours down to 10 minutes hands-on time. So while the automation of these library preparation protocols has allowed a significant saving in time and the costs, it also has additional benefits, including standardizing the results and reducing the biases that are inherent in some of particularly user biases, maximizing the high throughput and the total number of samples able to be run on a single run, and also reducing the footprint of the equipment that's required to actually run these. protocols. A study in 2020 by Tecan et al. Classified the degrees of automation in high-throughput sequencing into 4 different tiers. These range from pipette-assisting devices such as the Gilson Pipette Max in tier 4, tier 3, which are the liquid handlers, which are more open source in nature, such as the OpenTron system. Tier 2 describes the more dedicated commercial systems which run specific protocols such as specific library prep kits from particular commercial vendors. These include the Agilent Bravo and the PerkinElmer Cyclone. And then Tier 1, which are the most complex machines which run numerous protocols and different activities, not just related to high throughput sequencing. And one example of these is the Hamilton NGS star. The MagicPrep from Tecan Genomics is one of the newest Tier 2 automated liquid handlers. And it has a small footprint, which is, as you can see from the picture on the bottom of the screen, is able to fit into some of the most clustered lab spaces. The MagicPrep itself has a hands-on time of about 30 minutes at the beginning of the whole protocol, where the samples are loaded into reagent racks. Reagents are unpacked and loaded into the machine, and then the user is able to select the number of cycles and press start. Then they're able to walk away for an RNA-seq pipeline. You're talking about 8 to 11 hours when a machine performs the library preparation. And on a DNA sequencing pipeline, you're looking around 5 hours. And this is for 8 samples at a time. So in order to assess the effectiveness of the MagicPrep, we designed 2 small-scale experiments to compare its performance with those of other assays that are on the market. We looked at the RNA sequencing pipeline on the MagicPrep and the DNA sequencing pipeline. And for the RNA sequencing, we identified 2 sample sets, one from a pig and one from mosquitoes. We used 3 library prep kits to compare the QC factors between those methods from the reads that were sequenced from those sample sets. We then looked at the DNA sequencing pipeline in a separate study, and we looked at the sequencing metrics and the QC and some of the analysis components from a single sample that was had different inputs going into the DNA sequencing pipeline. And the 2 sample sets that we used, each was composed of 2 biological samples, which were run in triplicate. And the first set was from a pig, and the second set was from mosquitoes. The first set with the pig was an established organism in terms of generally it's the QC is relatively straightforward. There's a lot of options for processing those pig tissue, and in terms of transcriptome annotations afterwards for RNA-seq, the porcine transcriptome is annotated to quite a high level of accuracy. The second set with the Aedes aegypti mosquito, that's a bit more challenging, so it does pose a bit more complication in terms of sample QC. The transcriptome and the genome aren't annotated to such high degree of accuracy as seen with the pig. So as I said, we have 2 sample sets, and within each sample set we have 2 samples which are were separated into 9 replicates, and then each of those samples was then sequenced in triplicate on 3 different library prep kits. So we have Brandex, which is our usual go-to RNA-seq library prep kit. We compared that with replicates also sequenced. Library prep also performed on the MagicPrep RNA-seq pipeline, and also the Tecan Universal Plus kit, which is the manual equivalent of that which is run in the Tecan on the MagicPrep. We then sequenced all Brandex sequenced samples from both porcine and mosquito library preps on a single NextSeq 550 mid-output reagent run. And then we did same for each kit. So in total, we ran 3 NextSeq runs each with all the samples from the library kit from individual kits. So the first sample set that we tested was provided by the mucosal immunology group at Purbite, and this was derived from a Babahan pig that was infected with influenza A virus. Pigs are large animals. They tend to share several immunological and virological aspects with humans, and they tend to exhibit a similar pathology to infection in humans, and even down to the infection with the same viruses, both pigs and humans. Therefore, pigs represent an important animal model for disease under One Health. And in the terms of testing new vaccines and therapeutics, these will be applicable to both pigs and humans. In this particular study, we had a Babraham pig, which is a highly inbred breed of pig, which well characterized. This was infected with H1N1 virus, and we compared pre-infection and 4 days post-infection. And we were looking at local lung immune responses. So post-mortem, the lungs were washed out with a saline solution as a bronchoalveolar lavage sample. These immune cell populations in saline solution were then extracted and characterized using RNA sequencing to identify the gene expression within those cells, within the lung, as both mainly CD8 and CD4 T cells, and other immune cells try and clear the virus. Here's a quick snapshot of the QC, and the main thing to note here is that all the samples are high quality, as you'd expect from fresh tissue from a pig, and also in a high concentration for yield, which is great for RNA-seq. The higher the concentration, the more diverse libraries you see. So this is a graph that shows, if you look at the histogram on the right-hand side of the screen, these are the number of reads which align to the porcine genome after sequencing an initial QC. So the top 6 are the MagicPrep, the second 6 are our Brand X kit compared, and the bottom 6 are from the Universal Plus kit with Tecan. And these are all replicates of the same sample, don't forget. And as you can see here, generally, with the exception of the first sample on the MagicPrep, which produced almost 20 million reads, which aligned with the pig genome, generally the rest are comparable, and the differences were significant between them. For RNA-seq analysis, it's incredibly important to normalize when comparing between samples, and this eliminates any factors, including the MNA, which have any variation, including the significant number of reads in one sample compared to some others, and so on. So when we normalize all the samples, you can see here they all normalize really well with the medium of the box plots aligning with the yellow line. So normalization was successful. So the next step was to try and assess where those reads which map to the porcine genome were actually mapping. And the graph here shows, again, the top 6 samples that were sequenced on the MagicPrep, using sample prep in the MagicPrep, the second 6 using Brand X, and the final 6 using the Tecan Universal Plus kit. And you can see here that in light blue are reads mapping to exonic regions. And so the majority of reads in all cases of methods were derived from exonic or coding regions of the genome, which is what you'd expect. And they're all comparable, so the differences weren't significant. This graph here shows the coverage across genes where the reads were mapping. It's kind of like an average coverage across genes for each method. And as you can see here, generally what you'd expect quite an even coverage across each gene, across the genes, from each of the methods. And what we see here is that that's the case with the MagicPrep and the Tecan Universal Plus kit. Also, if you note that the variation between the replicates is also relatively small also. However, we see a different thing, a different observation in the Brand X kit, which actually there's a bit more variation between those samples, and also we see a 3' bias, actually, surprisingly, in those gene coverage at the 3' end of those genes. The biological coefficient of variation is important, where it gives an indication of reads mapping to genes between replicates. And where there's virtually no variation, that indicates that there's a high degree of accuracy of mapping and between those replicates. So you want a biological coefficient of variation nearer to 0 than anything else. And in this case, we can see here we have quite a low BCV value. So this is indicative of high degree of accuracy of mapping of those reads. And when we apply PCA analysis or principal component analysis or clustering of these samples, the samples clustered by day. So the samples on day 0 clustered together and the samples on day 4 post-infection with the virus clustered together, as you'd expect. However, interestingly, as you can see, there was the two Tecan methods did appear to cluster more closely together. Okay, so in terms of the number of genes, this volcano plot here shows the number of differentially expressed genes in the pig in response to influenza A virus infection. And this gives an indication of the number of genes which are either significantly upregulated, as in red, or significantly downregulated, as in blue. And these are generally the genes which will be followed up in the analysis and then validated by the methodologies. So in summary, given the reduced burden of the library preparation between the kits, the MagicPrep kit performed comparably to other library prep kit, other manual library prep kit methodologies. All the methods exhibited a good mappability to the reference genome and were derived mainly from exonic regions, as you might expect. The Tecan chemistry has produced a much more even coverage across the genes, interestingly, and much less variation. And the MagicPrep and the differential gene expression and the clustering, the two Tecan methodologies, again, clustered a lot more closely than compared with the Brand X kit, which may reflect similar chemistries that they utilize. Okay, so the second sample set that we used were from mosquitoes. Mosquitoes were included for a slightly different purpose in that mosquito RNA is difficult to extract. It's frequently quite challenging to work with to get extract in good quality and also in concentrations that would give good RNA-seq results. Generally, some of the normal QC rules, such as RIN numbers, don't necessarily apply to in our experience, to mosquito and other insect RNAs. And this could be a reflection of its AT content and some repetitive parts of its genomes, which can make the sequencing also challenging and the analysis challenging. The analysis is also challenging in the sense that the annotation isn't as advanced as that that's available with the pig and so on. So the project, now the sample we get gained the mosquito sample, the group that we gained the mosquito sample from, the flavivirus transmission and pathogenesis group at Pirbright, they were looking to assess the mosquitoes of a particular species to identify candidate antigens for recombinant vaccine work. So this is of their interest. So these are from live projects at Pirbright. And again, after sequencing, initial QC and mapping of those reads back to the mosquito genome, again, one sample in particular stands out on the MagicPrep sample 1, which produced twice as many reads as most of the other samples, which then maps back to the mosquito genome. Also of note is that one of the Brand X samples, you can see number 1, or sample 7, that produced virtually no reads, and that was excluded from any further analysis downstream from this study. Generally, all the other differences, there was no statistical difference between the methods. Again, normalizing the reads, all the reads were normalized successfully as shown here. And then when, again, we looked at where those reads were mapping to, a majority of them were in exonic regions. And again, we're seeing an increase in the number of reads which are mapping from the MagicPrep, but the difference wasn't significant with the other two methodologies. Again, looking at gene coverage, we're seeing the two Tecan methodologies produce quite an even distribution of reads mapping across the length of the genes, whereas again, the Brand X reads seemed again to have this 3' bias. The more erratic trace here is from the failed sample, so we can ignore that. Looking at variation within the data set, and the biological coefficient of variation was slightly higher than that seen with the pig data set. And again, although it was close to 0, it showed a little bit more variation between the replicates. However, when we looked at the clustering and the PCA analysis, what we were expected to see in terms of correlation between sample sets. The clustering between the samples wasn't consistent. This is possibly reflected in a much more complex data set from mosquitoes, a bit more variation, and also bearing in mind that this is a very limited RNA-seq investigation, where we're using almost a quarter of the reads we usually would to perform this kind of investigation. So just to sum up, the mosquito genomic material historically has represented a more challenging type of sample. Comparatively, rates of mappability between the methodologies were demonstrated between the kits, and the low biological coefficient of variation again provides some confidence in the mapping accuracy, where the majority of reads were mapping to exonic regions. And finally, the clustering of those groups was a bit more unclear compared to the porcine clustering samples in the porcine dataset. And again, this may be reflected in the limited number of reads that we used in this investigation. So we also assessed the performance of the DNA sequencing pipeline on the MagicPrep. And to do this, we used baculovirus, which has an insert from immunogenetic region from a bovine genome. This is an MHC region from a cow, and it's responsible for influencing the variability of antibodies which are produced. So this was used as a part of a vaccine research project identifying variation in response to vaccines in antibody responses in cows. We use this sample at 4 individual sample. We set it into 8 different replicates, and then we use 4 different dilutions, each dilution in duplicate, to investigate the impact of this input concentration upon the final sequencing metrics. So what we found is that generally, as you can see here, all the samples that were sequenced generally were about the same range of between 0.2 to 0.4 million reads per sample, which results, that's on the column on the right-hand side bottom, and then the column on the top left shows the percentage of those reads which aligned to the bovine genome to this particular region. And in all cases, irrespective of the amount of sample that we put into the reaction, about 91 to 93% of the reads we produced aligned. It should be noted here that one of the samples at 0.2 and QC, so it's all pretty similar and pretty straight. And this slide just shows some of the basic QC between some of those samples at the different concentrations. And you can see it's all pretty consistent between the different concentrations. But more importantly, looking at mappability of those reads that were sequenced. So we're looking at where those reads map to on the reference template in the analysis. And you can see that almost 80% of those reads are mapping back to the reference that we have. This is a coverage plot for all 4 dilutions across the reference sequence that we had. Although it's not overly clear, we can see some dropouts across this, and this is probably more than likely due to some of the repetitive regions within this sample to be sequenced. But generally, we've got good coverage for the majority of all dilutions tested. So just in summary, again, there was comparable sequencing metrics across the different sequencing inputs, varying from 100 nanograms all the way down to 0.2 nanograms. In total, so we were able to sequence difficult repetitive regions, which was demonstrated by the drops in coverage for some of those parts of that template. But when we compared the consensus from all those samples tested on the MagicPrep with those obtained from other methods, they all matched and validated. One sample, should be noted though, failed at the lowest concentration of 2 nanograms input. So in summary overall, the MagicPrep high throughput sequencing library preparation machine performed comparable to manual kits in this limited test. So generally, it should be noted here that the RNA-seq was much lower number of reads than we'd normally generate for a proper RNA-seq investigation. But generally, the sequencing metrics were comparable with other methods across both DNA and RNA protocols. The work with both common such as porcine, for example, or more challenging mosquito samples in genomes. And the MagicPrep was shown to perform comparably again. And the DNA protocol works well across multiple genomic inputs. Finally, bearing in mind the time saving of the MagicPrep protocol, the 30 minutes up front compared to 2 days of work with some of the other RNA, the manual RNA-seq kits, the MagicPrep integrated quite well with these methods. I'd just like to acknowledge some of the people involved in putting this study and data together. And the main bioinformatician who processed the RNA-seq work at Pirbright was Adrian Silesian. His picture's on the top. Noemi Polo and Ji Wu, their pictures underneath, are both members of my group who helped to put this study together. I'd like to thank Elmer Trillinian and the mucosal immunology group at Pirbright, and Kevin Mamminger and the flavivirus transmission and pathogenesis group at Pirbright for the RNA samples, and John Schwartz at the immunogenetics group at Pirbright for the DNA, the bovine MHC sample. And these are just some of the references from the presentation. Okay, thank you.